Hello and welcome. This is Bhaskar Napte from Pharma Growth Hub and welcome in today's discussion. We are going to talk about two important terms which are associated with the drug product. The first one is drug product assay and the second one is drug product potency. So what is the purpose of making the video on these two common terms? The reason is the ICH guideline Q2R2. Now, this guideline talks about analytical method validation, and if you look at the reportable ranges for drug product, you will understand that there are two terms used under the procedure. The first one is assay of a drug product, and the second one is the potency of the drug product. I would like to make you clear on to how these terms are correlated with one another and how they are different from one another. And I'm also going to make a video on a reportable range for assay and the potency. I will give the link for the next video in the description below. So in case if you want to understand, the range needs to be evaluated for drug product assay and drug product potency. Please go through that video as well. So let us now begin with the video number 1, where we'll talk about drug product assay and drug product potency. So what is the drug product assay? What is the purpose of the drug product assay? A drug product assay is primarily used to determine the quantity or concentration of the active pharmaceutical ingredient or API within the drug product. So it could be a quantity. It could be a quantity or it could be a concentration. What is meant by concentration? For example, in case of the oral solutions, you have the concentration maybe 5 mg per 2 ml. That is the concentration. Quantity may be in case of the OST or oral solid dosage forms like a paracetamol tablet containing a 250 mg of the paracetamol. And the paracetamol becomes the active pharmaceutical ingredient or the API. I hope you understand the purpose of the drug product assay that can define the quantity or that can define the concentration. Just to give an example for further clarification, if you are very new to the pharma industry, the quantity is the labeled amount of API per dosage unit. Means maybe a 20 mg of telmisartan per tablet. So this becomes the quantity. What is meant by concentration? So the concentration can be expressed as mg per ml, for example, mg per ml. Maybe 5 mg of drug X per 5 ml per 5 ml. So this is the way you can define the quantity or the concentration. So this is the assay value now. The 20 mg of the paracetamol per tablet is the assay value, which is representing 100%. This is representing exactly the 100% assay. So if I get the value of telmisartan exactly 20 mg, then I can say, oh, this is 100% assay. But if I get, let us say, 19 mg of the telmisartan, then how much will be the tablet assay? Just calculate and let me know in the comment box. So 90 mg can represent 19 divided by 20 into 100. So how much that is going to be now? Okay. So if you want me to calculate, let me do it quickly now offline. But if you get the answer, please put it to the comment box, right? So here is the calculator now, and 19 divided by 20 into 100. This result into a 95% assay. I hope you understand. And similarly, you can also calculate the assay for the concentration if the content of drug X is found to be 4.9 mg per 5 ml. So if you get the 5 mg per 5 ml of the drug, it is called as a 100% assay. But in case if the drug substance X is found to be 4.9, how much will be the assay of drug product? Can you calculate that? So please calculate and put the value into the comment box. The assay, drug product assay in case of 4.9 mg per 5 ml, where the 5 mg per 5 ml is the actual labeled concentration or the label content of the drug substance. Awesome. Let us move on to the next page now. And so this involves quantitative analytical techniques like HPLC, gas chromatography, UV to measure the amount of API accurately. This is just about what are the different analytical techniques can be used to measure the drug's product assay. What is the usage? Now this ensures the drug contains the correct amount of API complying with the labeled specification. What is meant by labeled specification? What is meant by specification or the labeled specification? So this is nothing but every drug product must have the specification, and the specification could be simple like description. It could be a very important test like assay, related substances, and any another physical parameter, right? So what is the specification for assay now? Let us say you have a tell me certain drug product, tell me certain tablet, and the specification is what? 95% to 105%. Means what? For each and every batch that you release, the assay should be between 95% to 105%. Means it should not be less than 95% and should not be more than 105%. So this is the labeled specification of the tell me certain API present in a tell me certain tablet. So once you conduct the analysis, you are also going to confirm that yes, whatever is the specified quantity, like 95 to 105% for this product, you know, this particular batch is meeting that correct amount of API. That is, our drug product has the labeled specifications, is meeting the labeled specifications, means 95% to 105%. So this is about the drug product assay. Let us now understand the second term, that is the drug product potency. What is the drug product potency's purpose? So the potency refers to the drug's ability. The potency refers to the drug's ability to produce a desired therapeutic effect. To produce a desired therapeutic effect at a specific dose, means the dose we talked about 20 mg of the telmisartan. So there is also going to be the dose of the biological drug product, and that drug product, that dose should result into a desired therapeutic effect. It's measure of the drug's biological effectivity, activity or the efficacy. So what is the important point needs to be considered over here? The potency of the drug product actually defines the biological activity. It is not about the chemical concentration. It is not about the only quantity of the drug substance. It is going to be most of the times maybe the proteins, that this is the concentration which is sufficient. Absolutely no. That could be the chemical assay. But to understand whether that particular protein, once administered, going to deliver the required biological activity, yes or no? So that is assayed, that is defined by the drug product's potency. That is defined something by the drug product potency. I hope you understand the definition of drug product potency now. So this refers to the drug's ability to produce a desired therapeutic effect at a specific dose. So it's a measure of the drug's biological activity or the efficacy. So it is just like what drug is going to do with the body. It is something about what drugs do with the body. Let us now understand the next point, that is methodology. So potency is often determined through biological assays such as in vitro or in vivo models that evaluate the drug's effectiveness in producing a therapeutic response. The therapeutic response could be in terms of creation of more antibodies, or maybe in terms of reducing the body temperature. So that therapeutic response should be measured maybe by in vitro or in vivo models. So let us now understand the example of potency in biological drug, because this term may be new to some of you, and to be very frank, I am also quite new to this term. The first, the term is now, the first example is monoclonal antibodies, or the MAPs. That is called as the MAPs in the short form. So what is the purpose of conducting the monoclonal antibodies potency? So their potency can be measured through assay like ADCC, that is antibody dependent cell cytotoxicity, and I think this is generally performed in case of the cytotoxic drug molecules, or could be CDC, means complete complement dependent cytotoxicity. Again, this also gets done for the cytotoxic molecules. So monoclonal antibodies are going to be like the proteins or the biomolecules. Here is the example. The potency of an anti-cancer monoclonal antibody may be evaluated based on its ability to kill cancer cell in vitro. Ability to kill cancer cells in vitro means in the, not within the any living organism, but outside the living organism. That is the meaning of in vitro. That is the meaning of in vitro. So what is the efficacy now in case if the drug substance is cytotoxic or anti-cancer molecule? It has to kill the cancer cell, and that ability of this protein or ability of the drug product under evaluation in terms of killing the cancer cell going to determine the potency of this anti-cancer drug substance. This is an example in case of cytotoxic drug product. Let us now talk about the vaccines. So how the potency of the vaccines can be determined? So the potency of the vaccine is often measured by their ability to stimulate an immune response. How our body is going to respond to the any outside threat, maybe the disease. So for instance, the potency of flu vaccines, example maybe influenza vaccines, may be determined by its ability to generate a specific antibody in the blood. The next is the hormone therapies. For replacement therapies, potency may be assessed based on the biological activity of the hormone. Let us talk about the insulin, which is having the capacity to reduce the blood sugar levels. So how this hormone, I mean how this insulin is going to help in reducing the blood sugar, that is going to define the potency of the insulin containing drug product. So where is the usage of the drug product potency? The potency testing is crucial for biological drugs like vaccines and antibodies, where the biological activity cannot be solely determined by the chemical assay or by chemical test methods. So that's the purpose of evaluating the drug product potency. So assay is used for quantitative measurement of the API in a drug product, ensuring the correct concentration. However, the potency evaluates the biological activity or effectiveness of the drug, is specifically important for the biologics. Thank you so much. I hope you understand the difference between drug product assay and drug product potency.